Welcome back to my channel. In today's video, I'll be discussing the DNA polymerases in prokaryotes. I'll be discussing the differences between three DNA polymerases which are found in the prokaryotes like E. coli and other bacteria. Okay, let's go into these differences. I'll be discussing the DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Okay, I'll be discussing the differences between these three DNA polymerases. The first point is subunits. How many subunits are there? Okay, how many subunits are there in these DNA polymerases? DNA polymerase 1 is formed from one subunit. It is only having one subunit. Okay, whereas DNA polymerase 2, it is made up of 7 subunits. Okay, we all know that enzymes are usually made up of proteins, right? They are ribozymes as well, but DNA polymerase is a proteinaceous enzyme, right? And so this enzyme, what it does, it it is having a number of subunits, it is having a number of polypeptides in it. Okay, and so DNA polymerase 2, it is made up of 7 subunits. Okay, and if I talk about DNA polymerase 3, which is the main DNA polymerase which is usually involved in the polymerization in the DNA replication, it is made up of more than 10 subunits. Okay, next is the relative molecular mass. Okay, you can see on the screen that the difference between the molecular masses in these three um, DNA polymerases, the relative molecular mass given here in DNA polymerase 2 column and in DNA polymerase 3, it is of the polymerization unit. Now, just take a moment and see that the polymerization unit of third DNA polymerase is very much molecularly big, right? It is having a relative molecular mass more than these two. Okay. Next is let us talk about some of the enzymatic activities which three DNA polymerases have. And first is the proofreading activity. Okay. The proofreading activity. When DNA polymerization is taking place by the DNA polymerase, right, sometimes it may happen that mispairing can take place. Okay, they can be misread, a base pair can be misread by the DNA polymerase, and it can then, after misreading of it, there can be a mispairing of it, right? So whenever there is mispairing, what happens? The DNA polymerase stops. It takes a halt. Okay, and via its 3' to 5' exonuclease activity, it chips off that particular mispaired base pair and then starts its polymerization activity from the starting. Okay, so we all know that DNA is replicated from 5' to 3' direction, right? So 5' to 3' direction. So the exonuclease is of 3' to 5'. Okay, it will chip off a mispaired base pair at the 3' end. Okay, now these three DNA polymerases all are having this particular exonuclease activity. This specific 3' to 5' exonuclease activity is termed as proofreading activity. Okay, next comes the 5' to 3' exonuclease activity. We know that DNA, this is the DNA, this is the 5' end. Okay, this is the 3' end. This is a template. Okay, 5' end. This is a template. So synthesis will take place towards this side. Okay, synthesis will take place towards this. That is 5' to 3' direction. Okay. We know that these enzymes from the last video I uploaded, that is, we know these enzymes, what are they? They are having they require a 3' OH, that is, they require a primer, which is usually a RNA in case of the prokaryotes, right? So this primer, it needs to be removed, right? I already told you that DNA polymerase 1 is performing that particular function. So it is having which activity? It is having 5' to 3' exonuclease activity. Exonuclease, I forgot to tell you that exo means from the end, right? There are two types of nucleases, exonucleases and endonucleases. Exonucleases, these are nucleases which are chipping off. They are removing the nucleotides from the ends. Okay, they can be from 3' end, as the proofreading activity can be 5' end as the exonuclease activity. And endonucleases, these are type of nucleases which usually cuts between the polypeptide, or I can say the polynucleotide, right? They cut between the strands. Okay, they are not cutting from the ends. Okay, so 5' to 3' exonuclease activity, it is only only present in the DNA polymerase 1. Okay. Next is, what is the rate of the polymerization? Like how many nucleotides can these enzymes insert during the DNA replication per second? Okay. DNA polymerase can insert 10 to 20 nucleotides, whereas DNA polymerase 2 can insert 40 nucleotides per second, whereas DNA polymerase 3, which is the main enzyme used in the polymerization reaction, can add approximately 250 to 1000 nucleotides per second. Just see the speed, I can say, right? The rate at which the nucleotides can be added. Okay. So next characteristic is the processivity. Okay, first I'll tell you what do this word mean, right? So processivity means, like, this is a DNA strand. This DNA strand is getting replicated. Okay, it is getting, this is a 3 prime and this is the 5 prime, and this DNA is a template. Okay, it's a template, is the parent DNA. So this is getting replicated. Okay, during the replication, it is getting replicated. During the replication, a particular enzyme, which is, suppose this is a DNA polymerase, okay, don't mind my drawing. This is a DNA polymerase, okay, which is doing its work. Okay, by processivity, it means that how many nucleotides can this DNA polymerase add before it dissociates from the DNA template. Okay, let me explain by the different characteristics of the DNA polymerase. DNA polymerase can add 3 to 200 nucleotides before it gets removed, or I can say detaches itself from the DNA template. Okay, whereas DNA polymerase 2, it can add approximately 1500 nucleotides before it gets detached from the DNA template. Okay, whereas DNA polymerase 3, it can add approximately, or I can say equal to or greater than 5 lakh nucleotides. After adding 5 lakh nucleotides, there is a chance that it will get detached from the DNA template. So via this characteristic, I can say that DNA polymerase 3 is having a very high processivity. This was the reason, the processivity was the reason which led to the conclusion that DNA polymerase 1 and 2 are not the main DNA polymerase enzyme during their discovery, right? DNA polymerase 1 was discovered very much earlier in comparison to DNA polymerase 2 and 3. So why this enzyme was not regarded, I can say, by the main enzyme which is replicating the DNA, because of its processivity component. Because after adding some nucleotides, like up to 200 nucleotides, it was getting detached from the DNA template. It had to be, we can say, pushed back towards the DNA template. So DNA replication will just halt, I can say. It will take a lot of time to replicate the DNA template, right? So processivity is the very main component, and it is usually provided by one of the components, one of the subunits in the uh, in polymerase enzyme. Okay, and here in case of DNA polymerase 3, if I speak, what is the processivity component is the beta clamp. Okay, and beta clamp is responsible for the high processivity of DNA polymerase 3. It can add up to greater than or equal to 5 lakh nucleotides, and then there is a chance it can detach from the DNA template. Okay, the last is a structural gene. What is a structural gene? That is a DNA polymerase 1 structural gene is poly A, and the DNA polymerase 2, it is having poly B, and the structural gene is same as in relative molecular mass. The structural gene is given of the polymerization unit only, and Pol C is the structural gene of the DNA polymerase 3. Thank you so much for watching my channel. Please like, subscribe and share if you like my content.